My name is Tim, and this presentation is about GPU GWAS and how we utilize NVIDIA GPUs to achieve a significant speed up in implementing genome-wide association studies. We're first going to talk a bit about the $1,000 genome and then describe the GWAS statistical methodology, and finally review the GWAS implementation. We're currently undergoing a rapid transition towards everyday genetics. The first sequenced genome was completed by the Human Genome Project in 2003 at the cost of $3 billion. And the cost came down to $1 million in 2007, and now in 2012, we're able to sequence the genome for $4,000. The reason for the rapid reduction in cost is due to the brilliant and continuous innovation in sequencing devices. Not only are today's sequencers much faster than the previous devices, they're also being made smaller and easier to use. The portable desktop sequencer was introduced in 2011. It is priced near $50,000. The new sequencers makes it easier for medical clinics to start sequencing a patient's individual genomic data. The Minion device is actually a USB sequencer that you can plug into a laptop. It is scheduled to be released later in 2012. This recent Nature article talks about how a small clinic in Pennsylvania called the Clinic for Special Children is currently making pioneering contributions to the practice of personalized medicine. While there are a number of clinics that have begun sequencing the patient's genomic data, the Clinic for Special Children have found ways of applying the newly available genetic knowledge to achieve a tremendous benefit in improving their patients' welfare. The article provided an example where the clinic reduced the cost of a bone marrow transplant because the sequencers detected a rare genetic variant indicated by the patient's immunodeficiency. The key to the problem was that the immunodeficiency was diagnosed early, very early in fact, so that the clinic was able to treat the immunodeficiency at its inception and reduce the cost from $500,000 to $12,000. This example can be a template for personalized medicine in the future. It shows how our knowledge of our own individual genetic sequences can lead to an immediate reduction in medical costs and also lead to an immediate improvement to a patient's welfare. The article mentions a large challenge we're currently experiencing in our march towards everyday genetics. Previously, when sequencing an individual's genome cost $1 million, the interpretation was relatively inexpensive. But with the advent of the $1,000 genome, however, we are suddenly experiencing a huge deluge of genomic information. This article identifies the problem as the $1,000 genome and the $1 million interpretation. The imbalance between sequencing and interpretation costs could be seen in implementing genome-wide association studies. Genome-wide association studies are a revolutionary new way for scientists to identify genes that influence health and disease. It scans the SNP markers across the entire human genome, seeking the genetic basis of diseases. Genes discovered from GWAS provides the foundation for personalized medicine. The first GWAS studies were conducted in 2005, consisted of 100,000 SNPs derived from 150 patients. The current studies analyzes 1 million SNPs from cohorts of 64,000 patients. GWAS via logistic regression is based on the following univariate model, with the goal of calculating the beta variables B0 and B1. Pi is the expected value of the trait given the genotype. X is the genotype itself. The null hypothesis in this model is given by B equals 0. The rejection of this null implies that there is a statistically significant effect of the genotype on the trait being analyzed. The statistical procedure for implementing a GWAS logistic regression is quite involving, but a majority of the computation is conducted via the beta update step. The step consists of a loop that calculates the new beta by multiplying the following components. The first component is the inverse matrix of the second order derivative, and the second component is the first order derivative vector. As you can see, a majority of these variables are matrices. The width and length of the matrices are dependent on the patient's cohort size. The beta update step is repeated for all of the SNPs in the data set. As a result, implementing a GWAS analysis requires a large amount of computational resources. It is exponentially dependent on both the SNPs being sequenced and the patient cohort size. The addition of covariates further increases the computational demand of a GWAS analysis. Covariates are clinical or demographic factors that have a confounding impact on the GWAS analysis. An example would be a GWAS conducted on lung cancer would be more accurate by the inclusion of the covariate for smoking behavior. The addition of covariates can provide many benefits to a GWAS analysis at the expense of increased computational costs. The following model describes the implementation of a GWAS logistic regression with the covariate Z included. The analysis for a large GWAS study is normally implemented on a computational cluster with a job queuing system to partition the GWAS analysis to the different servers. Departments from schools of public health typically supports a large cluster for genomic analysis. Some of the examples are the Enigma cluster from Johns Hopkins University and the biostatistics cluster from the University of Michigan. The clusters represent one possible way to cope with the data deluge. By logic, with each additional server, we are able to process and interpret more genetic data. But since each additional server costs around $3,000 or more, it does not address the challenge of the imbalance between the sequencing and interpretation costs. We would argue that NVIDIA GPUs are the key to addressing that imbalance, and that only GPUs are able to provide the cost efficiency to keep pace with the growing data deluge. This is because the computing GWAS is data parallel like much like 3D graphical applications. We have utilized NVIDIA GPUs to achieve a 10x improvement in implementing a GWAS analysis. The three key components for constructing GPU GWAS are PyCUDA, Genome, and CUDA-C. PyCUDA is an open source Python toolkit. It enables GPU programming with Python. It directly accesses the CUDA computation API. It is convenient and full-featured. Genome is an R library for GWAS analysis. It is compatible with the current GWAS data formats. It contains functions for all aspects of GWAS, and it integrates with PyCUDA via the rpy2 library. The dataset that we utilized is the publicly available Phase 3 data of the International HapMap project. In particular, we utilized the Central European subset consisting of 165 individuals of similar genetic ancestry and 1.4 million SNPs. This section describes the structure of the GPU GWAS. The program first reads the GWAS inputs from the HapMap dataset and then performs the GWAS logistic regression via a PyCUDA implementation. The majority of the work for the project was conducted in this section. The program had to incorporate the involving statistical methodology that was described previously. Numerous trial and error methods were attempted, and only after many sleepless nights was an efficient GPU implementation finally achieved. The results from the second step is encapsulated as a Genome data object and passed to R for further analysis. GPU GWAS was originally implemented on Ubuntu Linux. It is compatible across all major OS platforms. CPU analysis was performed on a Dell PowerEdge server with a dual Xeon quad-core processors. The analysis completed in 12 minutes and 18.79 seconds. And this is the CPU Manhattan plot. It returns a SNP on chromosome 7 as the most significant SNP. The results of the GPU analysis match the results of the CPU analysis. It completed in 1 minute and 18.9 seconds. The Manhattan plot also shows the chromosome 7 SNP as the most significant SNP. We're currently in the process of improving GPU GWAS. And the first step that was taken was to analyze the program with NVIDIA's visual profiler. The blue components are indicates GPU computation. We have identified the computation of the matrix dot product as a major point for improvement. We're aiming to improve performance by incorporating the PyCUDA sparse library, the CUDA library wrapper, and improving the memory transfer. We're in the process of integrating GPU GWAS algorithm to run on a GPU cluster. Since there are no communication between the GPU instances, we have found that each additional GPU translates to an 100% parallel efficiency. We have achieved a 10x speed up for a cohort size of 165 patients. while contributing to Professor Salih Jaffrey's cardiac research team at George Mason University, it is evident that CPU programs do not scale well with matrix size. From this diagram, we can see that the CPU Monte Carlo implementation of the calcium-induced calcium release model scales poorly as the matrix sizes grows larger. We have also found that GPU implementations scale better. The GPU computational time remains static even after n grows to 20,000. The implication to the GPU data parallel architecture is that we're potentially enabled to analyze larger patient cohort sizes and also enabled to incorporate more covariates. Implementing GPU GWAS will not only achieve computational speedups, but also could potentially enable larger scaled and more accurate GWAS implementations. We started collaborating with Professor Dahanjay Vadia from GenStar to analyze an experimental GWAS dataset. The dataset consists of 2.4 million SNPs from 2,000 individuals. We're looking forward to a substantial improvement of the program's performance. And it is also an opportunity to validate the GPU results in a scientific GWAS study. In summary, GPU GWAS is extendable to GPU clusters. It is integrated with an established GWAS library, and it has a potential speed up for larger data sets. Future goals include to encapsulate GPU GWAS as an R package available via the CRAN or bioconductor repositories, to implement a large-scale analysis on a cluster of GPU servers, to extend GPU GWAS to other widely used aspects of genetic analysis. A corollary to this project is the possible utilization of GPUs for clinical genomic applications. As we have shown, a single GPU can substitute for a number of servers, and the increased efficiency is scalable from small clinics to large hospitals. The GPU applications could potentially enable a distributed system of genomic analysis where each clinic is empowered to analyze the genomic data locally. This project would not have been possible without the great opportunities and support generously provided by the professors from both universities. I would sincerely like to thank Professor Jeffrey Solka and Professor Salih Jaffrey from George Mason University, and would also like to thank Professor Dahanjay Vadia and Professor Diane Becker from Johns Hopkins University. Thank you very much.
Thanks, Tim.